Good morning, good afternoon, and good evening, everyone, and welcome to the 8th webinar in the Cardiovascular Connections 2022 series. Today's webinar is titled Cardiac Tissue Slices, Preparation, Data Acquisition, and Analysis, featuring Dr. Bradley Palmer, Assistant Professor of Molecular Physiology and Biophysics at the University of Vermont. Today, he will demonstrate the challenging preparation of cardiac tissue slices and measurement of force and calcium. Before we get started, we would just like to acknowledge our partners at the American Physiological Society and the European Council for Cardiovascular Research, and particularly I'd like to shout out our session sponsor, Ion Optics, for helping to make this event possible. Ion Optics develops and manufactures data acquisition and analysis systems for cardiovascular research, principally functional characterization of excitation-contraction coupling. Their newest offering, currently in the final stages of development, will offer real-time examination of function in cardiac slices, including force length work loops and quantitative calcium. And now I'm pleased to invite Bradley Palmer to the floor. Brad, take it away whenever you're ready.
Welcome to today's webinar. My name is Brad Palmer. I'm a faculty member in the Department of Molecular Physiology and Biophysics at the University of Vermont. The outline of today's webinar is as follows. First, we'll introduce the cardiac slice as an experimental model system of the heart. We'll then demonstrate the preparation of cardiac slices from a rat heart, and then we'll walk through 3 functional measurements that can be made with the cardiac slice: intracellular calcium regulation, isometric force production, and mechanical work loops. So first, an introduction. A cardiac slice is a multicellular section of myocardium. This cartoon illustrates that the cardiac slice represents a section of myocardium usually prepared from the left ventricle. The slice is most often a few millimeters long and a few millimeters wide, and between 100 and 300 microns thick. With the tissue being so thin, the diffusion distance for metabolites and byproducts is quite short, and there's no worry of unintended hypoxias. We'll see more detail of cardiac slice preparation a little bit later in this talk. As an overview of its usefulness, the cardiac slice can be loaded with fluorescent dyes that allow for imaging of intracellular calcium, for example. The cardiac slice can be attached to a force transducer, which would allow for measurement of contraction relaxation function. And if the slice is also attached to a length motor, a force length work loop can be generated to mimic the pressure-volume relationship of ventricular cardiac cycles. So these 3 types of measurements that I'm showing you right here, calcium dynamics, contractile dynamics, and work loop generation, these 3 measurements will be discussed later in this webinar. But there are a few other methods that can be that are suitable. There are other applications that are suitable for this model system, and I'd like to present those. These other applications include studying multicellular signaling, such as action potential propagation and conduction velocity. Gene and protein expression can be examined, as well as biochemical processes and post-translational modifications. And imaging of cellular, intracellular, and extracellular structures and processes are also possible with the cardiac slice. The slice maintains the in vivo architecture of cells and extracellular matrix, so therefore the cardiac slice is advantageous for many imaging applications. And then finally, long-term incubation is an important use of the cardiac slice. There are some laboratories that have been able to maintain functionally viable cardiac slices for weeks and even months, which allows for examination of cardiac remodeling, for example. For the remainder of this webinar, however, we will not be discussing these 4 applications listed here on this slide, But if you're interested in references for these applications, you can look at the Resources tab, and you should find some helpful references there. Next, we would like to demonstrate how to prepare a cardiac slice from a rat heart. The first step will be the removal of the right ventricle. And you see in this still image right here, the base of the heart after having removed the atria and the valves. So we're looking right at the base of the heart. And the opening of the right ventricle is a crescent-shaped opening that is pointed to right here. And you'll see a little bit better, you'll see in the video how that arises. And while the opening of the left ventricle is the round opening that you see right here. So now we'll show this video of removing the right ventricle, and this video will take about 1 minute. So what you're seeing here is that the experimentalist is grabbing the right ventricular free wall with the forceps, and then just cutting away the right ventricular free wall with a small pair of scissors. The idea of cutting away the right ventricular free wall is to expose the septum so that the septum can be obviously visualized. So once the right ventricular free wall has been removed, what remains is the septum and the left ventricular free wall. But now with being able to visualize the septum easily, we can cut directly down the septum from the base to the apex. And with that cut made, the left ventricle is now prepared and ready to be sliced into the cardiac slices. So after the right ventricle has been trimmed away, the left ventricle is then taken to the vibratome for slicing. And the still image that you see here right now shows that the razor blade is about to be cut into the left ventricle. But what I'd like to show you now is another video of how to prepare the heart for this slicing in the vibratome. And this video will take about 3 minutes. I'm starting here showing you the ventricle as we had just previously prepared it on the previous video. We're going to put the heart onto this glass slide that's about 40 millimeters wide and about 25 millimeters deep. Here's a close-up, and what we're doing now is putting histoacryl adhesive onto that glass slide. And then we'll place the left ventricle onto the adhesive with the epicardium side down, and the endocardium is facing up. This particular adhesive is very quick acting, and it only takes about 30 to 60 seconds of holding the tissue to the adhesive, and it will adhere. And as quickly as you can, you would then put the relaxing solution onto the left ventricle. You'll see us skip ahead here in just a minute, and what's happening is we will put ice around the entire chamber to make sure that the relaxing solution stays cool, and we will also bubble the relaxing solution with oxygen to make sure that the solution around the myocardium stays oxygenated, and therefore the slice is more viable. And now I've sped up the slicing, but you can see that the vibratome is vibrating the razor blade back and forth and then moving the razor blade across the myocardium. The blade amplitude is about 1 or 2 millimeters, and the frequency is about 80 to 200 hertz. This is a more modern looking, I'm sorry, not just looking, this is a more modern vibratome that's being used here for creating cardiac slices. But it's the same general idea. In this case, the left ventricle was glued to a metal pedestal that is held in place with magnetically. And then the razor blade comes across and creates the cardiac slice. In this case, the cardiac slice is about 200 microns thick. After cutting all the way through, sometimes small threads of the myocardium may still be attached to the remaining LV, and you would just have to cut it away to make your cardiac slices. The question may arise then whether the slicing process destroys any of the cardiac myocytes. Several laboratories, including our own, have examined the viability of cardiac myocytes within the cardiac slice. The left panel here in this slide shows green for intact cells, which dominate the image. The red dots in this image represent strands of DNA that have been liberated during the slicing process. But take special note of the two arrows that you see in that image on the left-hand side. Those two arrows point to the absence of either color, which indicates there is an intact nucleus in a viable myocyte at that point. If a cell had been lysed or disrupted during the slicing process, the nuclei would be shown in stark red, like that shown on the image on the right. In this image on the right, all the cells have been lysed by detergent to provide this negative control image. We and others have found that the fraction of viable myocytes in a cardiac slice of about 300 microns thick is around 90 to 95%. So now that we've produced several cardiac slices, they must now be clipped for functional analyses. You see in this still photo right here, two examples of clipped cardiac slices. We'll see a video now of the clipping and loading process of a cardiac slice. When I say loading, I mean loading into an apparatus. And this video will take about 3.5 minutes. What you see right now is just simply a handful of slices that have been generated and put into a relaxing solution and kept on ice. See, what you're seeing here is a station where we can put a slice, visualize the slice, and then cut the slice. So we've put the slice on a glass slide that allows for visualization. And as we orient the slice, we want to orient the slice so that the fiber orientation is left to right. In this image right here, the fiber orientation is left to right. Now you might see some scoring that's going up and down. That's scoring from the razor blade. But the fiber orientation and the force generation is left to right. Having oriented the slice appropriately, we then use another razor blade to cut the slice into a square or rectangular shape. And after the slice has been cut into that square or rectangular shape, we'll put some more histoacryl down on in this case I put it on an old razor blade, and take away some of the excess solution so that we have clearer access to the slice itself. And then we'll pick up these clips, dip it into the glue, dab off any excess glue, and then just place it onto the slice on one end. Again, putting the glue on it, dabbing off excess glue, and then putting it on the slice. And again, it only takes seconds for the glue to adhere to the cardiac slice. And then we'll put this slice back into the solution. Sometimes it's advantageous to just make sure that it's not glued to the glass surface. Here's a close-up of the final product. You can see how the clips have been glued onto the ends of the rectangular shaped slice. So now, with the slices having been created, we're going to now load that into the apparatus. You can see the slice now over on the left-hand side of this image. And we're going to pick up the slice and gently lay it into the apparatus, where the clip is going over one hook and then the other hook. And if necessary, we'll have to adjust or finally place the clip over the hooks. And then we can adjust the length of the slice so that it will develop tension between the two hooks. And that is basically the clipping and loading of the cardiac slice into the apparatus. One of the last things to do as part of the preparation is determine the starting length of the cardiac slice. In the image on the left, you can see that there is a microscope objective sitting just below the slice. So I've put this apparatus on an inverted microscope. And with an inverted microscope like this, sarcomeres can be visualized. That is, if your slice is a little bit thinner than 250 microns, you should be able to visualize sarcomeres quite easily. And so you see an example of sarcomeres there on the right-hand side of this panel. In this example, the sarcomere length was set to 2.2 microns. Oftentimes you'll find inverted microscopes also allow for fluorescent imaging. And then in those cases, the slice can be loaded with a fluorescent dye, such as Fura-2 or Fluovolt, and an excitation light directed to the slice. In this example, a blue light is directed to the slice for excitation of the dye, and the emitted light is depicted as green. This green light is then directed to a detector, and that will allow you to monitor what it is that you're looking at with your fluorescent dye, for example, calcium. And you could also put a red light or some other wavelength other than the ones you've used for excitation and emission, and you can direct that red light to a camera to continue to visualize the sample. In this way, fluorescence can be recorded while also simultaneously monitoring sarcomere length with the camera. Many of you are probably familiar with recording intracellular calcium dynamics in isolated cardiac myocytes. In the example on the right, you see a single cardiac myocyte that has been loaded with a fluorescent dye that is sensitive to intracellular calcium. When the calcium is high, the light intensity is high, and the calcium dynamics are represented by the fluorescent light. A similar recording can also be made with a cardiac slice. For a cardiac slice, however, there is an issue of motion artifact to contend with. Motion artifact arises when the sample moves in and out of the field of view, which occurs during muscle contraction. This is to be expected for the cardiac slices. If you use a ratiometric dye, however, like Fura-2, the motion artifact can be minimized. In this example, both the numerator and denominator signals include motion artifact, which is highlighted in the circles. When the ratio is calculated, the motion artifact, which is similar in the two original signals, is mostly nullified. There's not a total elimination of the artifact, but the ratio is often clean enough that characteristics of the calcium transient can be quantified. This ratio transient can be examined for physiologically meaningful characteristics such as baseline ratio, peak ratio, time to peak, time to 50% return, and exponential tau, that is the time constant that describes the exponential decay in intracellular calcium as diastole begins. And that is basically how you would measure calcium dynamics in a cardiac slice. We can also record contractile force dynamics of the cardiac slice using a force transducer. In this image, the force transducer is holding the hook that's on the left side of the cardiac slice. Many of you in the audience, I'm sure, are familiar with measuring contraction of the isolated cardiac myocyte. In that case, the shortening and re-lengthening of sarcomere length is recorded as a measure of contraction relaxation function. And often the maximum contraction, or the shortest sarcomere length, is used as a measure of contractility. With the cardiac slice, the force transducer records isometric contractile force, and contractility is often taken as the maximum force per cross-sectional area. Although the shortening transient and the force transient are not strictly comparable, both can be used to quantify contraction relaxation characteristics. And those characteristics of isometric contractile force transient could include diastolic force, peak developed force, time to 50% peak, time to peak, and time to 50% return, and also an exponential tau. And there's one other assay I'd like to present today and really focus a bit on, and that is the force length work loop. See, the cardiac cycle can be mimicked with the cardiac slice by controlling the length of the slice during its contraction relaxation transient. This can be done using a length motor, indicated on this slide by the two-headed white arrow I have on the right-hand portion of the image. Specifically, if the motor shortens while the muscle is in systole and then lengthens while the muscle is in diastole, we can mimic the cardiac cycle. And so before I begin that demonstration, let's take a little closer look at the cardiac cycle. What you see on the left-hand slide, what you see on the left-hand side of this slide is an image of the left ventricular chamber, imaged by functional MRI. The light coloring indicates the blood volume. On the right-hand side, you see a Wiggers diagram that displays the left ventricular pressure and volume relationship throughout the cardiac cycle. So this audience is probably familiar with this relationship, but I would like to point out 4 phases of the cardiac cycle. When the contraction begins, the left ventricular pressure is too low to open the aortic valve, and the pressure in the chamber therefore rises without a change in volume, and this is called isovolumic contraction phase, or IVC, noted here. And when the left ventricular pressure reaches the pressure of the aortic valve opens, and blood is ejected from the left ventricle into the aorta. This is the ejection phase. And then when the left ventricular pressure drops below the aortic pressure, the aortic valve closes, and the myocardium relaxes without a change in volume. And this is the isovolumic relaxation phase, noted here as IVR. And finally, as the left ventricular regains its original shape and re-lengthens, there is a suction developed and blood moves from the left atrium into the left ventricle. This is the filling phase, which also includes the atrial kick in late diastole. If we plot the pressure against the volume, this pressure-volume relationship shows a cyclical loop. It's often referred to as a work loop because the area within that loop indicates the mechanical work generated by the pumping action of the ventricle. You can see the 4 phases of the cycle we noted in the previous slide. The isovolumic contraction is on the right-hand side of the loop and shows a rise in pressure with no change in volume. And then the ejection phase is at the top of the loop, going from right to left, where the pressure stays high as volume is reduced, followed by the isovolumic relaxation on the left-hand side of the loop, showing a drop in pressure with no change in volume. And then finally followed by the filling at the bottom of the loop, where the volume is recovered at low pressure. Presenting the pressure-volume relationship as a work loop like this can be useful in demonstrating the effectiveness or ineffectiveness of the heart as a pump. And let me give you an example of that. Compared to the normal work loop shown here in red, a work loop from diastolic heart failure, shown here in black, would demonstrate an elevated diastolic pressure during the filling phase. Because the change in volume looks relatively normal, this diastolic heart failure is often termed heart failure with preserved ejection fraction, or HFpEF. The work loop from systolic heart failure is shown here in blue, where it's clear that the extent of ejection is reduced compared to normal, despite the relatively normal extent of pressures. The pressure-volume relationship is therefore useful in describing and evaluating pump function. The cardiac slice can be used to mimic the pressure-volume relationship. And you see on the left in this image a cardiac slice attached to the force transducer and a length motor. The contractile force, which is shown on the top right trace, is continuously monitored. And the motor position, which is shown in the second trace on the right-hand side, is controlled to assure that the muscle shortens during force production and then re-lengthened when the muscle is relaxed. And you can see the result in the video that's going to come up next. So explicitly, there's a force feedback control system that controls the motor position relative to 2 force levels, the afterload and the preload. The afterload is specified as the force to be maintained during the ejection phase. And you can see this in the video that when the muscle shortens, so you can see that the motor is actually shortening the muscle during contraction, and it's shortening to the extent of about 5 to 10%. The preload is specified as the force to be maintained during the filling phase. And you can see that also in this video, that the muscle is re-lengthened, getting ready for the next cycle. In this slide, you can see 2 example cardiac cycles at 2 different, recorded at 2 different sarcomere lengths. Note here that tension is plotted instead of force. Tension is simply the force normalized to cross-sectional area of the slice. The red traces recorded with diastolic sarcomere length of 2.2 microns. And the green traces were recorded after stretching the muscle 1%, and after the preload and afterload were adjusted to correspond to the new diastolic and systolic force characteristics of the muscle. The resultant work loops are shown here. Again, the red trace corresponds to 2.2 micron sarcomere length, and the green trace corresponds to a 1% increase in muscle length. The slope of the end diastolic positions can be used as a measure of diastolic stiffness, sometimes called diastolic elastance, and this is noted on the slide as the ED slope. And the slope of the end systolic positions can be used as a measure of inherent contractility, sometimes called the systolic elastance, and this is noted as the ES slope. With this methodology and the use of the cardiac slice, myocardial performance through the cardiac cycle can be examined without a fully instrumented animal or isolated heart, and the slice offers control of sarcomere length and other conditions that are not possible in other experimental model systems. So to summarize, cardiac slices are useful experimental model systems for studying cardiac function, and it may be especially amenable to mimicking the pressure-volume relationship of the whole heart. The slice as a model system provides many of the benefits of isolated cardiac myocytes, such as allowing for fluorescent dyes to be introduced into the tissue. And slices, at least in our experience, are easier to generate and more robust than myocytes. That is, they can last longer. And slices have the added benefit of maintaining a syncytium of myocytes found in vivo, thus maintaining in vivo architecture and signaling between cells. Experimental results using slices are more likely to have physiological relevance. I have a few acknowledgements I'd like to go through. I'd like to first thank Matt Caparizzo at the University of Vermont for being an excellent colleague. I'd like to thank Fotis Patolis and Cesare Terracciano at Imperial College London for their workshop on cardiac slice preparation. And thanks to Ion Optics for sponsoring this webinar, and thanks to Inside Scientific and the American Physiological Society for hosting. And I'm going to leave this references slide up, but you can find this list of references in the Resources tab on your webinar interface. And now I'd be pleased to take any questions you may have.
Thanks, Brad. Okay. The first question that I will ask you, thanks you for your talk. How does one minimize tissue damage during vibrotome preparation of the cardiac slices?
One way that you can minimize tissue damage would be to just be assured that your razor blade is as level as possible. There are some vibrotomes that may not have control over the razor blade position very well, but there are some vibrotomes that do have that control over the razor blade position and is able to maintain that position well. But other than that, I think you'll find that the slices are quite viable, that is more than 90% viable. But that would be one way to, optimize your viability.
Great, that makes a lot of sense. This next question was submitted a little earlier on. You talked a lot about the left ventricle. Can the right ventricle also be utilized?
I suspect it can be, although I've never tried it. You'll see that the right ventricle is already very thin compared to the left ventricle. But we have removed the right ventricle and stared at it and wondered if we couldn't give it a try. We just haven't given it a try yet. But like I said, it's very thin, and so we were just afraid that it wouldn't be worth our while. It would be worth a try, though, I think, because these thin sections, they're only about, you can make sections as thin as 100 microns. And so I suspect you can make slices from the right ventricle.
Yeah, it might be something to add to your to-do list.
Right, exactly.
Okay, our next question is from Mark. Could you please provide details on the adhesive and relaxing solution that you used to fix the heart prior to slicing?
I could give you all those details verbally, but it's best for you to go to the Resources tab and check out those references that I have with a special asterisk. There you'll see all the recipes for the solutions that could be used for either relaxing solution or dissecting solution, or for recording solution. That is just a basic solution used for recording intact muscle. There are three references in particular that I put an asterisk next to that are, I think, especially good for that purpose. And then the adhesive is a histoacryl made by Braun. That's also, the information for that is also found in those references.
Perfect. Yeah. So I recommend everyone pop over to the resources at any time during this talk, and you can download that file. Okay. Next question. How many slices do you get from one heart?
It will depend a little bit on how thick you make your slices. But if you use a typical 300 micron thickness, you might get anywhere from 5 to 10 viable, useful slices from a rat heart. From a mouse heart, it's different. You might only get 2 to 4 from a mouse heart. But again, it will depend a little bit on how thick your slice is. And I didn't go through too much detail here, but you've got to be careful. The first 2 or 3 slices will have an awful lot of endocardium undulations and discontinuities. So the first 2 or 3 slices are almost always unusable. And it's only after you get past the endocardium do you start making these really useful cardiac slices. And of those, like I said, it's 5 to 10 for a rat and 2 to 4 for a mouse.
Okay, that's great. That makes a lot of sense. On a similar note, what would you say are the most critical steps in a slice preparation? And I suppose that would kind of be your opinion in a way. How do you feel? How do you feel is the most critical?
Solution preparation, I believe, is often the most critical. I mean, of course, you'll have to have enough practice that you have the hands and the dexterity and already figured out all the tools that you need. Once you've gotten past that, it's really the solutions. The solutions have to be of the highest quality to make sure that your preparations are consistently good.
Perfect. Okay, our next question here asks, how quickly should an investigator aim to clip and mount the freshly produced slices? Since they're no longer being actively oxygenated, I assume this must be done relatively quickly.
That's a great question, because I didn't mention that during the video I sometimes would discontinue oxygenating just for the sake of having a really good video and being able to visualize what's happening there. But you want to keep them as oxygenated, you want to keep them oxygenated as much as possible. But also recall that they're almost always on ice and they're almost always bathed in a BDM solution, so they have very, very low metabolism. So if they don't have oxygen for, I don't know, even 20 minutes, it's not going to harm them. We've been able to also keep these slices in the refrigerator overnight without being oxygenated overnight, and in the morning we can still collect force data from them. It's not as good as fresh, but it's still amazing that they are still they're still quite viable even without being continuously oxygenated. So that's what also I mean by robust. When I see these cardiac slices are robust, they can really you can expect performance from these cardiac slices that you would not expect from other model systems, like a whole heart. You cannot afford to keep a whole heart away from oxygenated solution for very long, but you can afford to do that with a cardiac slice if it's being handled properly.
Yeah, that is amazing. Okay, that was a great response. Teresa asks you, well, she says, thank you for this insightful talk. You mentioned that the contraction amplitude is calculated as force divided by cross-sectional area. Do you take into account the change in slice thickness? thus in cross-sectional area, as a consequence of the applied stretch? If yes, how? If not, why? Hopefully that makes sense.
Well, hopefully I understand the question fully, but we tend to just, because we cut the slice in a rectangular shape, we just use the width of that rectangle as the width of the slice, and then the slice that, the thickness set on the vibratome as the thickness of the slice. And we might want to adjust also for the fact that about 92% of the cells are actually viable, and so you can by just multiplying length times the thickness and then multiplying by the viability, say 92%, that would be considered your cross-sectional area, that is your functional cross-sectional area. And so that's the denominator used to normalize your force, so you have a really good estimate of the tension that could be produced by the muscle. We don't take into account any possible discontinuities in the thickness or the width, because we so far just haven't been able to observe it. We just haven't been able to observe a significant amount of discontinuity.
Okay, hopefully that answers your question, Teresa. Natalie's question says, at which temperature is the work loop acquisition possible?
We've done it at room temperature, and we've done it at 37 degrees C. We haven't really tried, and we've tried in between. We've tried 30 degrees C, but so anywhere from room temperature to 37 C, complete success in any of those temperatures. Perfect.
Next question. Interesting, the point about it being an intact I don't know what that says.
Syncytium?
Syncytium. But do you get issues with areas not stimulating quite the same way as others, perhaps due to variance in excitability or orientation of the fibers, etc.?
We haven't observed that. Now, we're not the kind of laboratory that examines action potential propagation, but there are other groups that have examined action potential propagation, and in their control cardiac slices, there's a nice propagation of the action potential throughout the entire slice. There may be occasions that, as they've shown in those other from those other laboratories, if there was an infarct, for example, there would be a buildup of collagen that might prevent the propagation of an action potential across the entire slice. That does occur, but if you've got an otherwise normal or uninfarcted heart, I think you could expect it to get completely activated.
Okay. The slices you make is transverse sections, and do the sarcomere vary in AP with T/transverse sections or cross sections?
Sarcomere length. We haven't examined that kind of question. That is basically a question of variability of sarcomere length with perhaps where in the myocardium you've taken your slice. That's an excellent question, and I'm so sorry I don't have the scientific answer to that. I think someone may even have that answer already, but I'm so sorry I don't have it.
Okay, that might be something else, something for them to look into a little further. Sam asks, can you use ischemic cardiac tissue slices?
Yes, you can use ischemic cardiac tissue slices, but as I had mentioned just a minute ago, you got to be careful. Wait, ischemic. I'm so sorry. We actually have. I was thinking infarcted. I still had infarcted on my in my brain. I'm so sorry. Ischemic, yes, absolutely. In fact, we've performed those kinds of studies where you will reduce oxygen availability or reduce glucose availability, reduce also the the withdrawal of byproducts. And so the tissue does become ischemic right there on the experimental apparatus. And it will also recover from ischemia too. That is, you can simulate ischemic reperfusion injury using this model system quite well.
That's incredible. It's interesting. Okay, can you use flash frozen heart to section and analyze, or does the tissue have to be fresh? I think it's kind of related to a previous question asking about the viability of the slices.
Yeah, I don't think you can expect to see force generation after it's been frozen. You might be able to get very good images from that. So imaging is certainly possible. But so far, what I've noted, what we've experienced and what I've seen other laboratories experience is that you can keep the myocardium on ice without it being frozen, and you can still transport it over a 24-hour period and still then make your slices and still expect to get reasonable force generation and viable cardiac slices from that kind of situation.
Okay. The next couple questions are kind of related, so I'm going to put them together. Obviously, you talked today about rat hearts. Somebody asks about whether it's feasible to use mouse hearts, what the technical limitations of that would be, and then how young could those mice be to be able to use those hearts?
That's a great question, because the mouse heart is about 1/10 of the size of a rat heart. But we have found we can use mouse hearts quite well. It's just that you get fewer slices. But I would not recommend having a mouse heart or using a mouse younger than 12 weeks. That would just be my recommendation. You can certainly give it a try, but you want the mouse to be as close to adult age as possible.
Okay, that's great. Would you say what the technical limitations could be, if there are any, other than just it's very small?
Yeah, that's really basically it. It's just that it's so small. You want to make sure that you don't lose any myocardium that would otherwise could have gone into a slice. So you might want to make thinner slices to begin with to make sure that you're not throwing away myocardium that could be incorporated into a slice. But you still might only get 2 to 4 or so viable slices at the end of the day.
Okay, great. Yan asks, why do the slices need to be 100 micrometers thick, or is that a personal preference?
It's basically a personal preference. I think they're anywhere, we tend to use them anywhere between 100 and 300 microns thick. I like to use them around 200 because then I could visualize sarcomeres. We can consistently visualize sarcomeres with 200 micron thick. We cannot consistently see them with 300 micron thick. So I tend to use the 200 micron thick.
Okay, great. The next question is wondering if you've ever tested this in atrium slices.
No, but that sounds like a great idea. I think along with the right ventricle, I think if as you develop experience with your vibratome, I think you'll be able to learn a way to to make slices from these other parts of the heart, whether it be RV or atrium. There's no reason why it couldn't happen. It's just, I'm sure, just a little bit more challenging than the LV.
Yeah. Okay, great. Can these sections be stained with fluorescent antibodies?
Yes, absolutely. So it's the slices, if I understand it correctly, they would have to be lysed in such a way that you can get the antibodies into the cells. But it depends on what kind of antibody you're using and what it is you're actually trying to image. But they're very useful for imaging, even with antibodies. Yeah.
Great. Okay, we are approaching the end of the hour, but I think we can fit in one or two more questions. So this person asked, you kind of mentioned before about the ischemic hearts. Can this prep be used on heart disease preps? And with that, are there any additional experimental considerations?
I think you just may have to be aware of what disease states you're actually examining. We've looked at disease states of dilated cardiomyopathies, for example, so they have poor contractile dynamics. And with the poor contractile dynamics, we did have to be a little bit more careful about how we generated work loops because there's just poor contraction. But we still could generate the work loops. And like I said, it was just a matter of working with your disease state and understanding your disease state and what it is that, how it is actually affecting the myocardial function and still being able to work within that context. I don't know.
Cool. Okay.
I don't know that I have more to offer with that.
All right. I think that was a pretty good response. I think I'm going to make this next one the last question. I'm actually going to kind of squeeze two in there and see how that goes. They're both about how long one has to wait for the relaxing solution before you can start measurements, and how long the slices are viable during an experiment, specifically with electrical stimulation.
So the relaxing solution usually contains BDM, which will make sure that the slices are not contracting while you're preparing them. But that relaxing solution, once you've put the slice into the apparatus and now it's bathed in a recording or an experimental solution, you will then also wash away the relaxing solution from the tissue, and that takes only minutes. In fact, you start seeing contractile force within seconds, but a total wash away will take about a minute or two, depending on the flow rate of your recording solution. And then the other question was about viability. We've been able to keep cardiac slices on the inverted microscope beating continuously at room temperature for 36 hours, and they're doing just fine for 36 hours. If we took a little bit more effort into trying to keep them a little longer term, we could probably do that, because those slices, if put in the incubator, can last for weeks or months. In fact, we've been able to keep slices in the incubator for a week, pull it out of the incubator, and it's behaving almost like it's fresh after it's been in the incubator for a week. So it's at least compared to, coming from a world of dealing with isolated cardiac myocytes, it was considered quite robust.
Okay, great. Thank you so much. Thank you for the insightful presentation, a really great Q&A discussion. Brad, it's been a pleasure to have you with us, and I hope you enjoyed yourself.
Thank you very much, Sydney.
Okay. And thank you to the audience for joining us live. In closing, we hope you enjoyed this Inside Scientific webinar sponsored by Ion Optics and produced in partnership with the American Physiological Society and the European Council for Cardiovascular Research. And we look forward to having you with us for the rest of the series.